Hello guys, welcome back. Hope you all are doing absolutely fine and studying well. So today we'll be continuing with our microbiology examination division series with our next topic of parasitology, that is Plasmodium, genus Plasmodium, more importantly the Plasmodium vivax. So let us begin. This is a quick read through of the text, so hope it helps. Genus Plasmodium. There are 4 species of genus Plasmodium: P. vivax, P. falciparum, P. malaria, and P. ol. They are protozoa causing malaria in man. These malarial parasites are worldwide in distribution. In India, P. vivax and P. falciparum are very common, but P. ol does not occur. Life cycle. The malarial parasites pass their life cycle in 2 hosts, that is man, which is the intermediate host, and the female Anopheles mosquito, the definitive host. Asexual development of parasite occurs in man and sexual development in mosquito. Human cycle. The sporozoite is infective form of the malarial parasite. These sporozoites are present in the salivary gland of female Anopheles mosquitoes. Man gets infection by bite of the infected mosquito and sporozoites are introduced directly into the blood circulation. Thus human cycle starts. It comprises of the following stage: pre-erythrocytic physiogony, erythrocytic physiogony, gametogony, and exoerythrocytic physiogony. Pre-erythrocytic physiogony. Before starting erythrocytic physiogony in blood, sporozoites undergo a developmental phase inside the liver cells. This phase of development is known as pre-erythrocytic physiogony. The sporozoites, elongated in spindle shape, become rounded inside the liver parenchymal cells. They undergo multiple nuclear division and develop into schizont. The size of schizont varies in different species and it contains 20,000 to 50,000 merozoites. The pre-erythrocytic cycle lasts for about 8 days in Plasmodium vivax, 6 days in P. falciparum, 13 to 16 days in P. malariae, and 9 days in P. ol. After completion of this cycle, the liver cells rupture and release merozoites into the bloodstream. Erythrocytic physiogony. The merozoites released from P. erythrocytic physiogony penetrate the red blood cells. They pass through the stages of trophozoite, schizont, and merozoite. Depending on the species of malarial parasite, there may be 6 to 24 merozoites in the red blood cell. The red blood cell ruptures to release the merozoites, which attack new red blood cells and continue the erythrocytic physiogony, repeating the cycle. In P. falciparum, infections erythrocytic schizonts aggregate in the capillaries of the brain and other internal organs, so that only rings forms are found in the peripheral blood. Differential features of various plasmodiums are shown in the table 54.4. Gametogony. Some merozoites of erythrocytic schizogony develop into the male and female gametocytes known as microgametocytes and macrogametocytes, respectively. They develop in red blood cells. These are sexual forms and are found in the peripheral blood. The microgametocytes of all 4 species are smaller in size. Cytoplasm stain is light blue and the nucleus is large and diffuse. In contrast, the macrogametocytes are larger. The cytoplasm stains deep blue and the nucleus is small and compact. Exoerythrocytic physiogony. The exoerythrocytic cycle resembles the pre-erythrocytic cycle. Some sporozoites, on entering into liver cells, do not undergo asexual multiplication but enter into a resting, that is dormant phase. The resting phase of the parasite is known as hypnozoite. After some period, usually. Okay, so these are the diagrams respectively. Erythrocytic stage of the different species of Plasmodium. We see first we focus on the vivax one, wherein the trophozoite is the ring form. Schizonts have multiple merozoites and enlarged RBCs. And in male gametocytes, we see these are the following ones. Okay, so differential features of the different plasmodia of man. So we'll be only focusing on the Plasmodium vivax since it's often asked in examinations, so we'll do it. Feature: Plasmodium vivax form in peripheral blood, trophozoites, schizonts, and gametocytes. Early trophozoite or ring form. It is 2.5 micrometer in diameter. Cytoplasm opposite the nucleus is thicker. Trophozoite, irregular amoeboid vacuoles present. Schizont, 9 to 10 microns in diameter, almost completely fills an enlarged erythrocyte. Merozoites, 12 to 24 in number. Gametocyte, spherical, much larger than a red blood cell. Female gametocyte, spherical, larger than male gametocyte. Cytoplasm stains deep blue, nucleus is small and compact. Male gametocyte, microgametocyte, spherical, smaller than female gametocyte. Cytoplasm stains light blue or pale blue. Nucleus is large and diffuse. Malarial pigments: yellowish brown. Infected erythrocyte: enlarged pale sharpness dots present. Age of erythrocytes infected: young. Duration of erythrocytic physiogony: 48 hours. Up to 2 years, hypnozoites reactivate to become schizons and release merozoites. These merozoites attack red blood cells and are responsible for relapse of malaria. Exoerythrocytic schizophrenia is absent in Plasmodium falciparum. Therefore, relapses do not occur in malaria caused by Plasmodium falciparum. Now we see the mosquito cycle. The sexual cycle of malarial parasite actually starts in the human host by the formation of gametocytes, which are then transferred to mosquito for further development. A female Anopheles mosquito during its blood meal from the patient ingests both the asexual and the sexual form of parasite. Only the mature sexual forms are capable of further development into the mosquito, and the rest die immediately. In the midgut of the mosquito, one microgametocyte develops into 4 to 8 thread-like filamentous structures named microgametes. From one microgametocyte, only one macrogamete is formed. Fertilization occurs when a macrogamete penetrates into a macrogamete. The fertilized macrogamete is known as zygote. The zygote lengthens and matures into an ookinete. The ookinete develops into an oocyst. As oocysts mature, it increases in size and a large number of sporozoites, a few hundreds to thousands, develop. Inside it, the number of oocysts in the stomach wall varies from a few to more than 100. The oocyst ruptures and releases sporozoites in the body cavity of the mosquito. The sporozoites are distributed into various organs and tissues of the mosquito. However, they have a special predilections for salivary gland. The mosquito is now capable of transmitting the infection to man. Pathogenesis and clinical features. Infection with Plasmodium causes intermittent fever, which is named as malaria. Incubation period is 10 to 14 days in Plasmodium vivax, Plasmodium falciparum, POL, but it is 28 to 30 days in Plasmodium malariae. The typical clinical features consist of febrile paroxysms followed by anemia and splenomegaly. Febrile paroxysms. It comprises of 3 stages: the cold stage, hot stage, and the sweating stage. In the cold stage, the patient feels intense cold and shivering. During hot stage, patient develops high fever, 40.6 degrees Celsius, severe headache, nausea, and vomiting. Fever ends and profuse sweating occurs in the sweating stage. The febrile paroxysm synchronizes with the erythrocytic schizophrenia of the malarial parasites with a 48-hour cycle of erythrocytic schizophrenia. The fever. Okay, we'll come to the cycles in very few short time. Recurs every 3rd day with a 72-hour cycle, and the fever recurs every 4th day. Plasmodium malariae. Anemia. After a few paroxysms, anemia of a microcytic or normocytic hypochromic type develops as a result of destructions of the parasite's red cells during segmentation of the parasites. Splenomegaly. After some paroxysms, spleen is enlarged and palpable. Plasmodium falciparum is the most pathogenic of the Plasmodium species infecting man. There are no relapses in Plasmodium falciparum infections, but relapses occur in Plasmodium vivax infection. It invades erythrocytes of all ages, young and old, but Plasmodium vivax infects only young erythrocytes. Complications of Plasmodium falciparum infections include the pernicious anemia and the black water fever. Pernicious anemia. Pernicious malaria is a life-threatening complication that sometimes occurs in the acute falciparum malaria. It is due to heavy parasitization. Various manifestations of pernicious malaria are grouped as cerebral malaria, algid malaria, and septicemic malaria. Cerebral malaria. It is characterized by hyperpyrexia, coma, and paralysis. Brain is congested. Algid malaria. It is characterized by cold clammy skin leading to peripheral circulatory failure. Black water fever, very important. It is a manifestation of infections with Plasmodium falciparum occurring in those persons who have been previously infected and have had adequate dose of quinine. It is characterized by intravascular hemolysis, fever, and hemoglobinuria. The exact mechanism of hemolysis is not known, but an autoimmune mechanism has been suggested. Parasitized erythrocytes during previous infections act like an antigen against which antibodies are formed. When subsequent infections and treatments with quinine occur, there is massive destruction of the erythrocytes because of antigen-antibody reaction. Black water fever has now become rare because new antimalarials have replaced quinine. Immunity. Immunity is species-specific, stage-specific, and strain-specific, and lasts only till malarial parasite infection remains active. This type of immunity is known as pre-immunization immunity. Laboratory diagnosis, very important. Microscopic examination. Microscopic examination of a peripheral blood smear is the most important diagnostic procedure in the malaria. Thick and thin smears of the blood are prepared on the same slide or 2 different slides. Blood for smear should be collected a few hours and a height of febrile paroxysms because the parasites are most abundant during this period. Blood is taken by pricking a finger prior to start of antimalarial therapy. For thick smear, take a drop of the blood on the slide and spread it in an area of 1 centimeter square. For thin smear, take a drop of blood at one corner of the slide and spread it with another slide so that the tail formation occurs at another end. Both thick and thin peripheral blood smears are stained with Leishman stain. These smears are examined under oil immersion lens. Malarial parasites may be quickly detected in the thick smear, but species identification is difficult. Thin smear is examined for identifying the species of Plasmodium. The parasites are more along the upper and lower margins of the tail of the thin smear. A minimum of 100 microscopic fields should be examined in each slide. All asexual erythrocytic stages, ring forms, trophozoite and sejons as well as gametocytes can be found in the peripheral blood smear in infections with P. vivax, P. malariae and P. ovale, but in Plasmodium falciparum infection only the ring forms and gametocytes crescent shaped can be seen. Multiple rings in an individual red blood cell with ecol forms is the diagnostics of Plasmodium falciparum. Sharpness dot in the red blood cells can be seen in P. vivax and P. ovale infection, whereas Morer's dot and Siemens dot are seen in the red blood cells with P. falciparum and P. malariae infection, respectively. Red blood cells are enlarged in P. vivax, but there is no enlargement of RBC in P. falciparum infection. Serological test, indirect immunofluorescence test, indirect heme agglutination assay (IHA), and enzyme-linked immunosorbent assay (ELISA) are the serological tests used for the diagnosis of malaria. DNA probes, they are sensitive and specific methods for the diagnosis of malaria. Treatment: chloroquine is used for treatment of acute malaria. However, resistance to this drug in P. falciparum is reported widely. Mefloquine is active against chloroquine resistant strains. Okay, so now we move on to that cycle which we had left for the continuity of this topic. So yes, we do the cycle now of the life cycle of malarial parasite. Very important. So the very first we begin with the sporozoites which enter in the man. One second. So this is the first one, the sporozoites. The sporozoites. Sporozoites enter in man pre-erythrocytic exoerythrocytic cycle in the liver, pre-erythrocytic and exoerythrocytic cycle in the liver, then the merozoites, then to the erythrocytic cycles in the RBCs, then we have the trophozoites, schizones and merozoites, release of merozoites into the blood, then we have the gametocony which is microgametocyte and macrogametocytes, they fuse into the mosquitoes in the mosquito to form the microgametocytes and macrogametocytes. Microgamete and macrogamete fertilize, forms a zygote, ookinete, oocyst leads to sporozoites and the bites of mosquito. By the bite of mosquito, the sporozoites enter into the man. So this is basically the cycle wherein first we have the sporozoites in the entering into the man, then pre-erythrocytic and exoerythrocytic cycles in the liver, then merozoites which are formed are released. Erythrocytic cycles in the RBCs take place, which leads to the formation of trophozoites, schizones, and merozoites. Then the release of merozoites into the bloodstream, which leads to gametocony, macrogametocytes, and macrogametocytes into the mosquito form into macrogamete, macrogametocytome, and microgametocytes. They fertilize further to form the zygote, which further makes the ookinete, which forms to oocyst, which leads to sporozoites formation, which are stored in the salivary gland of the mosquito, and when the mosquito bites, they release the sporozoites into the men. Okay, so with that said, we have completed the topic of malarial parasites. If you do like the video, please leave a like, comment and share it with your friends, and also let me know which topic to do next. Thank you.